your domain of expertise. Schizophrenia is probably one of the most exciting fields in neuroscience, with psychosis and understanding how the brain can compute those states. So thank you very much for coming with us and talk to us about your work today. We appreciate that. I will leave and let you the stage. Okay, thank you very much.
Okay. Well, anyway, I'm 15 minutes only, and then we have 40 or half an hour to talk. So anyway, I have very long titles. So I want to talk about the objective discovery approach. So hopefully by the end, and how it relates to functional brain imaging. So here I have a couple of quotations from Busaki. It says, subjectively defined terms are, this is the problem he's saying about functional neuroimaging, or about neuroscience, but I will be talking about functional neuroimaging. Subjectively defined terms are selected as independent variables, and their dependent brain variables are searched for. These terms are inherited from philosophy rather than derived from objective investigation of the brain itself, or assumed to be real entities. So the point is, do we want to use the terminology that philosophy, he says, but also cognitive psychology or neuropsychology has bequeathed to us as ways to investigate the brain? Is that useful? He says, no, you have to do an objective discovery approach, which involves actually starting with measurements of brain activity and then working up towards what concepts those tell you that you measure. So he suggests that neuroscience should establish its own vocabulary based on brain mechanisms. So for my own research, this I agree with completely. Yet, this are some of my papers here. What am I doing? I'm doing papers on working memory, lexical decision, evidence integration. Those are all concepts that were given to me by psychology or cognitive psychology or neuropsychology. So how do I get out of this? I can't stop doing these. These are like 2022 papers. So at the same time, in the lab, we actually do objective, generating of cognitive processes from the data. So I'm transitioning out of this, but it's very difficult to sort of talk about new words that are not already bequeathed to us from other fields. So we have to establish our own vocabulary. So the goal for our lab anyway is to anatomically identify brain activity in macro scale networks, as well as their objective cognitive function. But the problem is, that is a very substantial problem. The human brain, or more precisely, human brain activity is not just one thing we can measure. Each functional brain imaging method is limited to its own specific measures afforded by different types of brain activity. So we basically, for me, I didn't have an opportunity to intracranial record. But if you're talking about measuring macro scale networks, that's the goal of our lab. Intracranial recordings are not going to work. First of all, like we learned yesterday, this is always done on the diseased brain. And secondly, how many thousands of neurons can you really measure that method? I have here, if you could measure 5,000, you're measuring like about the probability of winning the lottery. It's 0.00006% of brain neurons. So we're not going to be able to discover macro scale networks that way. So we really have to choose between MEG or EEG or fMRI. And so, like Sam's talk yesterday, gave us a really good introduction to what MEG or EEG measures. And the challenge is going to be, well, now we're actually directly measuring some aspect of neural firing. But like to actually, on a macro scale, be able to identify which frequencies are correlated or coordinated in a certain way is very, very difficult. Because even though we like to talk about theta, alpha, beta, gamma, and 4 bands, really there's kind of infinite number of bands of frequencies that are possible. And how do those work together, and how do they coordinate, and how do those networks actually piece together? That's a difficult problem. I don't think it's intractable, but it's very complex. And when you're talking about neural activity, you have 86 billion neurons probably inhibiting each other in certain ways. Very, very difficult. So luckily, fMRI is available. And this, of course, is influenced, the bold signal blood oxygen level dependent signal, is of course influenced by neural activity, but it's not a direct measure of neural activity. So in some ways, that's actually a relief. For me, it sure is when I'm trying to, measure macro scale brain networks. I can't deal with like the complexities of 86 billion neurons. But the bold signal is a very robust, simple measure that if we're doing what Busaki says, we allow it to tell us what it measures. Is it telling us what neurons do? No, it's not. It's telling us what bold signal does, which of course is pushed around by the need for oxygen from neurons, but it's so much simpler than 86 billion neurons and the frequencies they're firing at. I was just lucky that fMRI was what I started with because that allows me to, have a simpler problem. So what are the principles that we use to actually find these networks? These are the 4 principles. First, use a finite impulse response model because that allows the true human dynamic response pattern to emerge from your data. And so far, we've had a day and a half of talking about brain imaging, but not one person has shown yet, and I will show hemodynamic response to events. And in fact, we were at Human Brain Mapping in Glasgow a few months ago, and not one person showed it there either. So that to me is shocking that we, with the EEG, you see people will display the activity that you're actually measuring, but with MRI, it's always the picture or it's a conductivity matrix. It's almost never the actual elicited bold signal from some type of task. So first, finite impulse response model will allow us to identify that. We want to use individual voxels if possible, because if we're going to find networks, if you parcellate in advance, you're forcing certain voxels to be together on the same network, which is not going to be the natural state. You won't be able to identify the natural networks. A voxel has to be able to participate in multiple networks. You have to use a dimensional type of approach. You can't have borders because that's not the natural state. It's not like this voxel is on one network. That voxel could be on 2 or 3 or 4. The brain will tell you. Just ask Busaki. And then we need timing information because we want to have a function associated with our networks. And that function is best interpreted by objective measures, which those objective measures are how the bold signal responds to tasks, or responds to some type of timing. It doesn't have to be like a working memory task. It can be internal events like now I'm mind wandering, now I'm not, or any other type of event you can get timing. But if we don't separate out the variance that's related to that timing information, the networks that we observe may not be interpretable because, well, resting state networks typically are interpreted by just looking at the anatomy. But what we really want is a hemodynamic response shape associated with every network. So if you can take out the variance that is related to the task timing first, every network will be related to the task, and you can interpret them better that way. So following those 4 principles, we still analyze working memory data. So this is a paper published in Cortex in 2020, but it just sort of shows an example. In this situation, 4 networks emerges, and we interpreted them based on working memory ideas, right? So this peaks first, this network peaks second, this network peaks third, and this one's kind of in between. So we said, well, this network's involved in coding, this is involved in maintaining, and this is involved in response. And you can kind of see from the neuroanatomy that that's not bad. And then the default mode, well, it just deactivates sort of in the middle. So you can kind of say we got encoding, we got maintaining, we got response, and we got default mode. Hey, we're finished with working memory. Okay, maybe. But the problem is that network, the same network, emerges all over the place. Exactly the same network. So it sure as heck isn't this is what we call maintaining. But what we do in our lab when we're not publishing stuff that sort of uses the terminology from cognitive psychology is that we look through every single moment that network emerged, and we try to make up an interpretation for what that network might be doing, not based on top-down type of ideas, but based on bottom-up evidence from observing the human dynamic response shape, which is so rarely actually observed. So in this case, the same network. So here's when there's 6 letters in a working memory task is red, and when there's 4 letters is blue. When there's a delay, you get much more activity. This is a 4-second delay is dashed. So when there's a 4-second delay in 6 letters, you get much more activity when there's 4-second delay in 4 letters. And that's much more than even when there's no delay. So that's fitting in with the idea of maintaining information for working memory. But this other task has to do with silently saying to yourself the function of an object versus hearing the function of an object on a sound file. And we have much higher activity in this network when we're silently saying a function to ourselves than when we're hearing sound file play. So is that working memory? It's not working memory the way Badley would have had to think about it. It's more generating silently or attending to internal representation. And then this is another task that Donna Addis published, I think it was early 2000s. And this involves imaging the past and future. So in other words, thinking about what you might do in the future, imagining something in the future, imagining something in the past that you didn't actually do, and then recalling some information, or that control condition was actually creating a sentence out of 2 words. We find the same network. Now these, I'll talk about these networks in a bit, but anyway, here we have, when you're imagining the past, imagining the future, or when you're creating sentences, there's more activity in this network over the whole period when you're doing this compared to recalling. So now we have to think about that. Why is it that there's more activity in this network and more sustained activity in this network, imagining the past, imagining the future, making up a sentence, relative to recalling something? So think about that. That's what we do. That's how we objectively, come up with a function for these networks. So anyway, I have lots of examples. I'm not sure how I'm doing for time. Pardon? Okay. Well, okay, I'll just go through a couple more. Okay, so here's another network. So this network has a different function. This is not the same network. Okay, and this network is active during the incongruent condition on a Stroop task. It's actually switching between color and word. But when there's incongruent, it's active. When it's neutral, it's not active. And also when you're integrating disconfirming evidence, it's active relative to when you're integrating confirming evidence. And we've seen the same network multiple times, and it always peaks after response. Now we have to integrate all that information. What is this network doing? We're not going to use a term like integrating disconfirming evidence or Stroop. We're going to have to come up with a word that's independent of those. The word that we came up with so far is reevaluation, because it actually peaks after people are responding, which you would never see if you assumed a human now response. After people respond, we see this network active. It's more active in disconfirming than confirming. It's active in incongruent, Stroop, and not Stroop. What's it doing? Got to come up with a word. Here's a network that's involved in lexical decision on one hand, and it's very active when you're actually seeing a real word, but it's actually suppressed when the word is not a word, but it looks like a real word relative to some other conditions. So active when you're actually seeing linguistic information, but suppressed when you have to suppress that linguistic information. The same network is also involved in when you're discriminating an emotion in a face. So here's a scrambled face, nothing. Foil, which is sort of still a face, but it's not the correct emotion that you're looking for, and target when you actually find the emotion you're looking for. So all this has been published, but we haven't sort of integrated all of this information into a publication to say what these words are. We call this extraction of meaning. It could be linguistic meaning, or it can be emotional meaning. Here's another one. I won't bother going through that unless I think I might run out of time. But basically, this is what we call focus on visual features. The same network, it's completely active in this type of Ravens task, where you have to see what the next thing is here. So it's like, what would come here with dot, dot, 2 dots, and then 2 dots. I guess that's the right answer here, 2 dots. So, it's more active than that, but then electrical decision is actually suppressed. So what's about this visual network? It's active in this task where you have to pay attention to visual details, but it's suppressed when it's electrical decision. It doesn't really matter what the word is like. What matters is whether or not it's the word. To actually suppress it under some conditions, activate it under other conditions. So anyway, we've been spending the last, like everyone's talking about what they did during COVID. This is what I did during COVID. Look at all of these networks, find specific slices that actually are, unique to one network and not to any of the other networks. So we ended up with, I have here 12, but actually these two are the same. One is just one-handed response and one has a two-handed response. And so we use methods to explain the variance in the bold signal. And this set of networks explains all the variance in the part of the bold signal that's predictable from task time. And what's left over is just basically movement or some type of random noise. So far, in the tasks that we've looked at, everything we've collected, shared with other investigators, some downloaded online, these networks actually explain the task-related variance. but the words were all the work. What words do we use? And that's done by seeing when the network is actually active in every single task and then coming up with an interpretation, which is not obviously an ongoing process, because I think we've probably looked at 200 or so manifestations of different networks. And you know, there's basically probably five to 10 really good examples of each network in multiple tasks. We've gone through all those tasks, but I'm applying for funding to do a much larger project that involves more sharing and more online. So here's a reminder of what these principles were. A finite impulse response model will allow the hemodynamic response to emerge. Instead of assuming a shape, the smallest unit of measurement should be an individual voxel and not a partial or an a priori region of interest. Each voxel can participate in multiple networks, a dimensional approach, and we need timing information, some types of timing vector. So here's the process that we used. So first, we extract the networks and shapes. Sorry about the zoom. But we extract these, the network, and we can extract shapes. So anytime we do an analysis, we get those two things. Then we have to classify these networks. So I'll explain in a sec how we did that. But now we have these prototypes that are based on, you know, many manifestations of these networks. Then we interpret the shape, and what we're working on now is actually associating the activity in these hemodynamic response shapes, so individual differences in the level of activity with individual differences in other traits. But so we're sort of just not finished that. But just quickly, I think I'm out of time. So just quickly, this is like there's only 1 or 2 slides left, but I just want to quickly say how we classify this. So here's the to be classified image, and here are the exemplars that we've developed. We just vectorize. Now we have critical slices. There'll be sort of 178 critical slices will actually distinguish all these 11 brain networks. And then we just string them out into a row like that. And then we take that from all of the exemplars, look at the correlations, and then the highest correlation will tell us which is the best match. And it may be that sometimes there's mixed, because the principal component analysis is not perfect. So I don't have time to explain the whole thing, but this is my last slide. So anyway, Saki says we need to establish our own vocabulary. So I think that we haven't established the vocabulary, but I think we have a methodology that works, which is find these networks. That was possible with bold signal. With MEG or EEG, it'll be much more complex. The time scale is much faster or more precise. And you have, you know, multiple frequencies. They have to be able to identify these networks is difficult. There may be far more networks than fMRI has. And then intracranial recording. I mean, I've never had an experience with that. I'm not sure what types of networks you'd be able to detect that way. So anyway, that's kind of my proposal for what the approach could be. And I guess the discussion can be about whether that's a valuable approach and what types of combinations of these might be fruitful. But anyway, the bottom line is the bold signal was just a lot easier and it was very rewarding to work with. But our lab's moving to EEG now, so it's ready for the next challenge, I guess.
Thank you very much, Todd. I think like this is, I don't think students can appreciate how courageous it is to advance those kind of idea and change completely the vocabulary, because we're not only changing the vocabulary, it's kind of like reversing the history, because in some way we all started in cognitive neuroscience from the psychology concept and try to put images on them. So I really like this idea. More and more, like actually my lab is doing this and kind of like questioning about like those framework in which we try to put those images in that don't really fit and how we can explain those. And definitely going with EEG and MEG for me too. Okay, so we're going to go with Rebecca and then Sam.
Todd, yes, 2 questions, one sort of more methodological and conceptual. So methodologically, isn't the like the sluggishness of the bold response? You've said in the past that the bold response is really useful because it allows you to fractionate, you know, as a given number of networks, a certain taxonomy, right? Identify a certain taxonomy that's limited.
First of all, there's not multiple. mean, there are
Yeah.
you can measure frequency,
Right.
of course, resting state people do. But when you're actually using task-based MRI, the shapes that are produced by the old signal in response to the signal are so simple. I mean, you saw that, you know, they're complex enough to spend years studying them. But that's, I think, one of the major kind of saving graces of using the signal. It's just very, very
So
simple.
the pushback you've got, people say, oh, well, how can you just identify, you know, however many number of networks, have 9 or whatever it is. When the complex, you know, when the brain networks observing all these different complex cognitive processes are so diverse, how could you just have that? And so your answer would be, it's just the simplicity and the crudeness in a sense.
Well, it's the whole point of the talk, which is that we're not measuring neural activity.
Right.
We're measuring neural activity, we're even duty. That's difficult. You want to come up with networks for 86,000 neurons, it's impossible. But EEG makes it possible. But with fMRI, it gets easy.
I'm just trying to sort of grasp what it is about the bold signal that makes it sort of tractable.
Well, I mean, it was a bottom-up thing for us. I didn't know that it was going to be this easy. But it turns out that when you actually take the task-related variance out first, and then you just let the data tell you what the bold signal is instead of assuming it. I mean, there's infinite shapes that you'll see of the bold signal, but the surprise was that the anatomy is reliably in each task. You can actually find a small set of networks in every task that we've done so far. But you know, maybe when like not all brain areas are graded eliciting the bold signal either. So it's not like, again, this is not a great study of the brain and the neurons in the brain, but it is a great study of bold signal. A great way, I think, to study bold signal, which is not the same thing. So when we have more powerful magnets and we have faster TRs and we can actually get into those brain areas that are not that graded, eliciting a bold signal, we would probably find more networks with fMRI, but this is the most basic, fundamental kind of set, I think, that, you know, that we can at least, we should understand that before we build on it more.
The second part of my question, very different. So you started with the Bizaki challenge. So do you think of cognitive processes differently? As somebody trained in cognitive psychology, has the bold response changed how you think about how cognition works, or how to categorize it?
Well, for me, it's kind of made me move into a space that actually is separate from cognitive psychology. And that space actually gets to be more like neuropsychology. So I went to the University of Victoria as a graduate student. That's where my PhD is from. And that's a clinical neuropsychology. It was then, and it still is a clinical neuropsychology school. So that was all about assessment. So we're going to have people do this test. And clinical, clinical neuropsychology can avoid these questions about causation, about complexity. It's just, let's see how the brain is doing. Let's measure this behavior and compare it to norms. So to me, fMRI gives us a way to measure some type of thought processes that are somehow in between neuropsychology and cognitive psychology. They're more general than cognitive psychology, but they're more specific than clinical neuropsychology. Because in clinical neuropsychology, you'll get a score and it'll be like this executive functions, right? This is, you know, some type of memory, whether it's working memory or whether it's solid memory. And so, but this is, you know, look at that one working memory experiment. We had 4 networks, all of them with different levels of activity. So if you know, could say, we have a set of networks here, we can actually have a norm for how every network responds to every type of task. Let's say you've got, say, a battery, a matrix battery, one for schizophrenia. So you could actually norm each network in that battery to some extent. So you have a side of kind of like a new, potentially a new neuropsychology. So to me, it's no longer about lexical decision and working memory. It's about these new cognitive processes that fMRI is giving us that you couldn't get anywhere else. And so can that help us assess the patient? Can that help us understand delusions? Can that help us know how to stimulate the brain to affect, you know, a certain type of behavior? So yeah, that's.
I think that's great. Sal?
Yeah, I mean, I hope I'm interpreting your talk correctly. I agree with you and Buzaki that we're kind of inheriting things from philosophy. And when you try to do a basic subtraction image, if you say there's attention and that's one atom of thought, network, memory. There's one sort of atom of thought. There's your network. But those aren't the real sort of networks. If
The
you're
ones that Bo would give us.
not the ones that but I mean, all of our tasks are also inherited from that those assumptions too. So I mean, to really get at what are the, you can't do it with an fMRI, but like to really know what are the recurring eating network configurations of OLD or EEG, wouldn't you almost need to have someone walking around with like naturalistic interaction with the world and then see when pattern X repeated and then what was that person doing? Is there anything common about that person? And try to essentially hit the history eraser button on psychology and just say, there is no such thing as lexical
you're right,
decision about it. It doesn't exist. Then everything else is not tied to a specific identifiable thing that we could name after we erase history is either part of, endogenous unconscious processing, that type of network, or it's something to do with some level of time lag inherited from processing of past events or predicting, modeling future events.
I lost you in the last sentence there, but let's just talk about the first part and then maybe the second part. So the first part, do we need to have people walking around? I don't know about need, because we've learned quite a bit using the tools that we had available by trying to investigate cognitive biology. But those 4 principles are what matters. So as long as we have timing of when people did things, then that's a great way to go. And maybe if we use more naturalistic, types of timing information, we would have different networks.
I guess what I'm saying is if you're having somebody do all of the tasks that I use and that you use are inherently carrying the baggage of philosophy that's cognitive.
agree with you.
So, I mean, if I say I'm going to go back into all my data and find things that recur across all of my tasks, I mean, I'm finding commonalities in the common baggage of cognitive psychology, which is, I think you're right, but I think the problem is almost deeper, where you have to assume, the lexicon is wrong, the tasks are wrong, the ideas that, the separation of cognitive processes into attention, memory, episodic memory, semantic memory, that's all wrong, and start from somewhere more fundamental in the brain and its sort of spontaneous processes and interaction with the environment. Because the task, I mean, if cognitive psychology's baggage that needs to be purged to get at the real proper lexicon of the brain, it's also in the tasks, right?
So I agree with you, but we got fairly far with all that baggage to the point where at least we have the methodology. And if you sit there and say, no, it wouldn't really work because we're using tasks from cognitive psychology, you kind of lose the opportunity to take advantage of what we already have. But it would be better to completely not use those tasks and have naturalistic type of environments where you can get both signals when people are walking around, or maybe a virtual reality, or whatever it is. But the data is just not available. Like what we do have is actually quite a few types of tasks. Like one of the tasks that we analyze from the Human Connectome project is this task where they have little shapes and boxes, and the shapes either interact socially or they move randomly. Analyze that, which I guess is this much longer type of thing. You're watching a movie. That's not a real, not naturalistic environment, but it's not the typical type of task we use. And in addition, a lot of the tasks that we develop in our lab have to do with schizophrenia and measuring delusions, and they're not the standard types of tasks like that evidence integration type of task. So we do have quite a range, but you're right. I'd much rather if the project was all naturalistic fMRI and that I had the money to collect over the next 10 years all the data we're talking about. Maybe we will. Maybe that will be possible.
Can I just ask a quick follow-up to Sam's? And that is, how would you parcelate, or maybe it's a question for you, how could you possibly parcelate all the experience that someone has walking around in order to get anything out of that? Like, don't you need to have some sort of repeating thing that people do so you can average things together?
Yeah, I mean, yes. I guess what the way that don't give up.
It's not what you think.
I guess the way what I'm arguing is to really do what Buzaki says is I'd have to be wearing an fMRI and you'd have to see, okay, how many times are there recurring patterns? And then you'd say, okay, there's a recurring pattern. What was Sam doing at time X, Y, and Z when that pattern recurred? So that it either had something to do with something I was doing around that time, like reaching with my left hand, or it would have to be something endogenous to the brain's own self-maintenance or internal operations, or it would have to be time lagged. Like you can't tell by looking. Sam's like, crap, I need to respond to that email. And you would figure that out. It's difficult, but you'd have to, I think what Buzaki is saying, it might be right, is that the entire way that we parcelate the mind is at least partially wrong. It's baggage from philosophy. And if you were to do it the other way, you'd have to do something, what I'm recommending. And I'm very much in favor of what Todd's done. Don't get me wrong. I'm saying you purged half the baggage, but the tasks are in themselves the other half of the baggage, right?
Yeah, but I think like to come back to the question, and like the issue is like if you look at the literature, like between 60 and 80% of studies that are published in a high impact journal cannot be reproduced. That maybe is the way it's testing, and like and also one of the big findings in psychology is like if you put somebody in the dark room closed, he impacts his cognition negatively, and his performance is extremely diminished, which is kind of an fMRI, right? With somebody else with noise, and we put in like white down to. So I to go even further on that part, like for me, like I really think that if we continue to use those constructs, but not even at a philosophical level, but at the experimental level, without asking those challenging questions that Todd is asking for us today, we're going to repeat the same thing all over and over. And I'm sure you have example in your field that might have scientists that have done the same work in 40 years, never asking that question. It's like am I measuring release of brain signal in fMRI, or like is that really the working memory task I'm doing? Am I really measuring working memory there? Like is it really working memory with them? Because it's so. This is not a task that nobody would ever do in their life outside of the class. But like we denature the pathways or mechanism completely, and we accept because what we're trying to is reduce the noise. So I think like it's time, at least for me in my lab, we're trying to push on the other way, saying like, how can we go more and more towards a natural way? Like, so it's showing, like, for example, focusing more on EEG, focusing more on MEG, trying to get away from the causal part and really trying to work on try to get away from this challenge. I'm not saying that's the solution. I'm just saying that this is the hypothesis we're growing. Okay, yeah.
Can I just jump in really quickly? Follow up. Sorry, Danielle. I feel like the debate of your. The follow-up is like, okay, so Buzaki makes the point, and others have made it that like our cognitive constructs from William James and philosophy don't necessarily reflect what the brain is doing. Point taken, but isn't that like the cognitive constructs that we work with are no good, or they're not useful, or is it that they're useful? And the problem in cognitive psychology and cognitive neuroscience is we treat that they're heuristics, they're useful categories that we've made up to describe these families of phenomena that we sort of model with experimental tasks. But is it that they're wrong, or that we reify them? We treat them as if they're things in the world, and then we go and do experiments to find these things that we've made up. And to me, that's more the problem than that these tasks that sort of isolate these cognitive processes, tell us something useful, and again, gave the foundation for Todd to sort of do a more data-driven look at cognition. I don't know. To me, like those do we necessarily have to throw out all of cognitive psychology, or do we acknowledge its limitations?
Yeah, I just didn't want to say to throw out cognitive psychology. I just think that if you're going to study fMRI, then you have to let go of cognitive psychology.
And Sam has done that.
I think one of the factors, we're gonna end up having you have the moderators that. I think one of the things that came up today, actually, from your talk, is indeed that we're assuming that there's an isomorphism between the cognitive construct that we're measuring and the fMRI signal. And that's a really bad assumption, and that's even an assumption in your analysis. You came the closest to actually checking for that isomorphism with your analysis. You don't know what the function is between cognitive constructs that have been studied for measurement. They have some validity in terms of predictability. So there is something. You can't say the brain doesn't produce memory. Otherwise, what is it? Your foot? No, probably it's your brain. But it's the question. We don't know how that transformation is actually done.
Bold signal. The brain, of course, produces memory, but what does the bold signal actually detect? That has to do with memory. That's really what I'm talking about.
I think you're wrong. The question is the transform between the bold signal and the actual cognitive construct. You don't have it in your analysis.
Okay, so wait. Okay, which part of what I said was wrong? Because I said that memory is a thing, which I agree with you. And then I said the bold signal doesn't pick up, can't pick up memory, which it does respond to the process of.
Sure, but you don't have anything in your analysis that tells us how to transform from the bold signal to the emergent feature that is the cognitive construct.
Yes, what I have is looking over hundreds of tasks and seeing how the bold signal responds.
That's
to those tasks.
we're getting back to this.
Or to what Sam suggested.
Getting back to the
So wait a minute, you're saying you can never put any behavioral label on a bold signal construct?
I'm saying think a bit more how you get from the neural signal to bold signal to the cognitive construct. Not doing that, you're just predicting things.
I don't really get.
There's no mechanism what you're looking at. The closest we come is what she's or doing a generative model.
Well, okay, so wait, Danny's like, yeah.
This whole time and I have nothing to say anymore. On that, I was gonna say what Sam was gonna say, and I'm done. I'm not coming in on the space, but I'll ask a question later. What basically I was building on what Sam was talking about, which was like one point in your talk, I was sort of thinking like, where do you go next with this? And you had a correlation with openness to experience, and I was just interested in is that. Like, how do we link those bold patterns now to behavior? And are we, by taking something like openness with experience, are we creating a similar problem on the other end? Or is it okay because that term was derived the same way that you're deriving the terms by data-driven and then giving a label to something, which is what people did with the big five. So is it okay because you're doing that, or would it be better to just give all the individual items and see what shakes out in a PCA or something? So yeah.
Well, so you're basically asking, okay, well, there's a couple of like whether it's possible philosophically or whether it's practically possible. And the practical side is you have to come up with individual differences in hemodynamic response, which is what, of course, fMRI. So first of all, we have individual. Just to focus on what Randy and I disagree on. He's saying, I think, that you can't put a word to that.
But
But you're saying that's impossible.
it's just that's a correlation. That doesn't tell you actually how this is correct.
Okay, and with your question, it doesn't matter what word we put on. It doesn't matter. Because if you're okay with just the anatomy, use that. Just look at the anatomy. Okay, so you actually have individual differences in the bold signal. And whether or not you want to put a word on what that bold signal does, or whether or not you want to just look at the anatomy, you can correlate that with any set of individual differences and other measures.
Yeah, so my question was about those things.
So-
And are we like, we've put labels on those behaviors that were or personality dimensions, and there's a whole historical context for how those were derived. And my question was, are we in trouble there too? If we ever, I hear Randy's point, and my question's actually removed from that. It's more just like, we want to connect, whether we agree on how we do it on the brain side, but now we've got a network that we want to correlate with a behavior. Are we in hot water on the behavioral side? And this can be the BFI or the depression inventory or whatnot? Or are we okay there because the same methods were used to derive the openness to experience construct or whatever it may be? I just sort of wanted to hear your thoughts about that.
So there's the independent variable side that I already talked about. You're talking about the dependent. variable
Exactly,
side.
yes.
And so the method that we use to find these task-based brain networks is the same method, of course, that we would use to predict individual differences in any range of variables from individual differences in hemodynamic response. And so those, of course, would just get thrown in the way you did with all the boxes in the brain. And then what you're doing then is taking individual differences in whatever measures it is, personality, cognition, whatever, and constraining the variance in that set to that which is predictable from brain activity. So now any components that you get out of that will be related to individual differences in the brain activity. And so you can go through the same process where you can try to label those components
Right.
based on whether it's a combination of personality, cognition, or gender, or age, what it ends up being. So it's again an atheoretical exploratory.
Right. Yeah, which I think is a little different from what you had on your slide and is preferable, right?
Right, yeah.
Well, regarding Randy's point, and maybe this is what you were saying already, but I'm thinking of what you're doing as an exercise so that you can then do traditional experiments but with better categories, better tasks or something like that. Isn't that a value where like maybe you don't care initially what the mechanism is, but you can do better experiments if you're basing your tasks on clusters of brain activity.
Maybe.
I'm trying to help you here. No, but I don't that's your point, right?
No, it's not really my point, but I guess he's asking, how are you going to do different experiments now that you have different networks to worry about? I really thought that, more focused on what Danny mentioned is to find out whether or not there's a correspondence with behavior, with the activity, individual differences in the activity. It's not so much about doing more fMRI experiments. It sort of lost the fun for me when I realized I was getting the same networks from all different experiments.
Yeah, that's wrong.
This is a less controversial. Unless you want to jump in.
No, go ahead.
It's actually more to do with what you're doing seems to be similar to what Russ Woodward and colleagues did with Neuroscience.
It's okay, well, that's a great question. So what Russ Woodward did with Neuroscience is say, what we have to do is we've got these words like working memory and attention, but those are not the right words. Those are not the natural, the natural cleave nature at its joints in terms of word cognition. So what we're going to do, we're going to look at every task that we can find. We're going to find out how they overlap, and then we're going to give new words, right? So all it's doing is making the same problem, because before we had some words and now we have more words. So now we're going to, it was his solution to reverse inference, where we can't keep kind of like giving these words like working memory to brain activity. We need smaller words so we can link that up.
I thought it was more like different, looking at towards intersections and see if there's a different way of labeling.
Well, it's just a different type of basically psychology between set of words.
No, because it seems more to what you're doing, because you're using psychological driven tasks to identify commonalities.
Well, if we're going to give labels to networks, we can forget about that. We can forget about that. We disagree on that, so people can decide for themselves whether they think that's possible. But really what we're doing is forgetting about all those words and just finding out what networks there are, right? Just let it tell us, and then let us tell us what the networks, what the hemodynamic response shape is for all those networks. That's kind of it. Words are more fun. Like maybe there isn't a word, maybe there isn't a good word, maybe it isn't possible. But I'm not going to sit there and look at the way this network activates over 200 tasks and then not come up with some theory about kind of what it might be doing. I mean, that's kind of like what we do as people who are interested in human behavior. So, but anyway, I just wanted to make the point that I don't think what Paul Direct did is a solution. I think it's just leads to the exact same problem. And what we need to do is go from the bottom up, find out what the networks are, and then build it up from there. Okay.
I have a boring methodological question about the way that you like quantify whether it's the same or a different network. And I think we've talked about this a little bit before, but can you unpack a little bit like your rationale for choosing those slices and then computing, I think you said you do a Pearson correlation versus I was envisioning something more like a dice metric where you can take like a 3-dimensional shape and see like if those actually overlap, like the percentage of overlap. Because I worry, I keep going back to like, I don't know, my field is memory and people are really interested in like, okay, in this task, you get like this hippocampal subfield and this one, you get this hippocampal subfield. And like, it's such a nuance, like these voxels versus these voxels. And maybe we're doing things the wrong way, but it feels like, I don't know, it's something more precise to say like, this is the network, this is the same network. It doesn't veer like a little more ventral in this particular task. And that veering is like, there's meaningful information there. Like how similar are the networks? Is it like 90% overlap, 95%? I just want to hear your thoughts about that.
Well, I mean, a number is kind of what we're trying to, I can't give a number, but like at first we started to do that with the whole brain, but it didn't work because there's so much overlap in the visual cortex. So I kind of gave up on it in a way that I did this for years, basically a way that ended up with only me being able to classify the networks, me as a person, because I knew the patterns and I just couldn't train anybody to find those patterns. So eventually, like this is what I did during COVID, is I just found the patterns and I, have sort of burning out my eyeballs on a laptop screen and I have, 6 or 7 or 8 different of these networks open. I go to the slice and I find, okay, that one's always in, when that one's there, also that one's there, and also that pattern there. So we went through and found probably about 5 to 10 patterns for each network that are only there in that one network.
You were your own little machine learning algorithm.
Yeah, because that's the thing with computer. A correlation couldn't do it because the whole brain was just like too much variation, but it ended up being patterns that we took out and we actually drew on the slices and gave names to that only our lab knows about because they're all stupid names. They're all silly names like jumping jack flash and bilateral eyeball sitters and stuff like that. And no one from outside the lab has ever asked to see it or has even been interested in really how it's done. But anyone trained in my lab gets to know that just as well as they get to know any other. So that's how we did it, is like, I just, then once someone in my lab, Chantal Percival and Sahi came into my office and said, why don't we just do correlations on only these slices? And I'm like, oh, that never works because we've done that with the whole brain. They go, but we tried and it does work. So you have to check it afterwards visually against the manual that has all the shapes in it. But if you use those slices that are the important ones, the critical ones for distinguishing between networks, and that works that way.
Well, Daniel, I think that you're asking also the question that Todd was referring at the start. It's kind of like we are prisoners of our own framework.
Yeah.
Like, in memory, so for me, I started my career at UFT, which is a huge department on memory. And what about can I have like powerful enough fMRI to really map this campus and see how it gets there. And that comes from the fact that memory has been always like that, right? Like dividing and trying to
Absolutely.
compartmentalize those concepts. I think like at least in the approach that Randy and Victor and I are trying to push forward in some ways is to get away from the concept, which would be very hard in your field because I don't know how your reviewers will feel about that. But and that I think is one of common points of what I'll say today.
I just forgot to mention another part about your question, which is that it's not the actual box like they're here or a few over. It's the overall pattern.
Sure. Yeah.
Yeah. So it's like, those catch butt things where we click something, we can see a lamppost or whatever. It's like that. Like a computer can't really tell the pattern, but so
Yeah, and like your point is fair. Like one of the reasons I brought it up is because I fear that that's a criticism that you would get. And I've asked you about this before because of people from my field and related fields who think that way. And it's hard to say.
On my paper.
Sure, yeah, and like I'm sympathetic to both sides, right? Because there's really cool stuff happening in those fields, but that's sort of, and like being trained that way also in Toronto, that was the first thing that kind of came to mind when I was reading through Todd's work, and we've had discussions about it, and so I fear that that's a criticism that could come back, and I think the nuance of how you say this is this network is something that's not trivial for somebody to get the buy-in for. So.
Yeah, and it's for me, like it's also at the technical level, you know, we come back to that when we since this morning, I'm trying to general discussion there. Is this when you go to the detail, when you collect data, you see how much movement you have. And how's the step in fMRI we have to go through to normalize our data and to talking about voxel, it's like no sense to me, especially when you know that there is migration of neurons and like, I mean, like, and so I think there is an inherent limit that back to what you were saying today about the technology that we use when we talk about fMRI, go toward those questions, and we kind of like block our mind saying, okay, let's forget that this limit and saying like a voxel is really that, so I'm going to talk like that when I'm interpreting my data. I think this is very dangerous, and that's where we go with what I was saying, answering with Dylan, is this notion that you know, a lot of our studies are not replicated. You know? So like we're talking about 60–80%. I mean, this is gigantic, and those are the best studies in the field. So even if we have a significant level, I think this is very important to ask those questions that we're asking today instead of like, you know, like what is our framework? Are we really studying what we think we are studying? And there is this approach of reductionism, right? Like we're going to reduce the object we study as much as we can, like that we get rid of the noise. I think for me in my past is like I try to do this with clinical trial methodologies. And what I found at the end of the day is like I'm not satisfied with my 40, 100 subjects that I have there. Really doing what I'm trying to study. And then I get another study that kind of like not really dependent. So I thought like this is really kind of like probably this generation of scientists that try to push that on the other way. We have faith also scientists. This is not precise enough, and they are right. I mean, you know, but I don't see a way out, at least in my data, in MEG, in fMRI, that if we go toward the precision, we're going to have a sustainable answer that can be repeated over time. That's the dangerous part. Sorry. So thank you very much. So that was it. And the morning and thank you. Thank you so much. And thank you for the.
I don't know what you're